and for the form and the function of the organism. So my answer is no.
Thank you very much. I believe we're at the time. Is that correct? Or you have 2 minutes? 2 minutes.
I want to say the last sentence about my splash of milk. Who is afraid of a splash of milk?
Who is afraid?
Of a splash of milk.
Oh. Well, thank you very much, Dr. Samanti, and Mr. Garani Gary, it's your witness.
Thank you so much for being here, and I certainly wished I could speak Italian because I think you have some fascinating things to say, and I've tried to take some notes, and I appreciate all of the things that I've heard you say.
Thank you.
I don't think we have any questions, Dr. Samanti, but thank you.
Thank you. May I go?
Yes. Yes. Thank you so much.
Thank you so much, and arrivederci to everybody.
Our next and last witness for the day is Dr. Ralph Seelke. Ralph? Dr. Abrams, members of the committee, I'd like to introduce you to Dr. Ralph Seelke. Dr. Seelke, would you please introduce yourself and provide a brief background of your qualifications to testify today about the Minority Report?
Okay. Thank you. Again, my name is Ralph Seelke. I'm a graduate of Clemson University in my bachelor's degree. My PhD is in microbiology from the University of Minnesota. Actually, my work was at the Mayo Clinic, however. I then did postdoctoral training. I went from bacteria to chickens at the Mayo Clinic. For the last 16 years, I've been an associate professor or professor in the Department of Biology and Earth Sciences at the University of Wisconsin-Superior. So I think I'm the cheesehead that was referred to, the mistaken cheesehead, although I live in Minnesota. So, but I have to be very guarded on my support for Packers and Vikings. It becomes a serious issue. My primary duties, I'm primarily an instructor. My primary duties involve teaching courses of a cellular and molecular nature. Microbiology, genetics, immunology, and cell biology are primarily what I teach. I've been following the conversation in the U.S. about the teaching of evolution for a number of years, and that resulted in actually an active research interest in evolution. Since I was trained as an experimental biologist, when my interest was piqued by things like Darwin's black box, I immediately turned to experimentation. And my interest has been in determining by experimental methods the capabilities and limitations of the evolutionary process. And because of that interest, I do try to stay informed about the research literature in experimental evolution. So I'm primarily here to speak about the use of microevolution to produce inferences on macroevolution.
So how does that work? I mean, talk a little bit more about your experimental work.
Oh, I'd love to take more time. Basically, what experimental evolution does is we use experimentation to ask very specific questions about evolution. And this comes from the fact that primarily in bacteria, there are numerous examples of evolution. And if you want to show my little PowerPoint, you could do that. But I actually had a table of some of the things. That's not it. There's probably a dozen or so very well documented cases of evolution in bacteria, and typically those cases involve a gain of function where you're able to make able to use a food source that a microbe in the past was not able to use. And so you can do real experiments with evolution. In a day, you can produce 1 trillion organisms. So you can ask questions. Now, 1 trillion organisms is a lot of organisms. When you talk about human population, we're talking orders of magnitude more than the number of people who've ever lived on this planet. You know, since the beginning, wherever you want to determine that. So you can ask bacteria to do very, very specific things and find out, can they do it or can they not do it? So experimental evolution, and you can model time. One of my heroes is Richard Lenski at Michigan State, and Richard Lenski has been evolving bacteria for 35,000 generations. He does this. It's actually his technicians do this, but he takes 2 teaspoons of bacteria and he grows them in a medium that's low on glucose. And then after they've grown, he takes a 100th of that population and puts it in a new tube. You get 6.64 generations every day. He's been doing this for 15 years. You do the math. With a little work, you can model trillions of organisms and literally tens of thousands of generations. 35,000 generations puts you in the range of what human evolution, what is supposed to have happened in human evolution. So experimental evolution allows you to ask real questions about what evolution can do.
And by any chance have that on a memory stick?
I have it on a CD, but
do you have the CD?
Let's see if it's over in the. It was basically what I had as a chart of successful and unsuccessful evolution experiments that happen. And by doing this, there's a number that are successful. There are a number that are in fact unsuccessful. Now, I'm rather proud of that chart of unsuccessful evolution. It is not easy to find. People don't tend to flaunt their failures. But I have been able to produce a chart of 8 or 10 cases of evolution where people looked very hard for evolution to take place, and it didn't happen. And so this is the sort of thing that can be readily done in this. So yes, you can. So the chart was just showed some examples of successful evolution and some examples of unsuccessful evolution.
Can you describe for me in more detail an example of unsuccessful evolution?
Well, one of the things I'm doing now is one of my other heroes is Michael Behe, and Behe said that if you have multiple independent events that have to take place, you will simply not be able to observe evolution. And so last year at this time, I was a visiting scholar at Stanford University, and I basically built some molecules. I made some changes in a gene, and I put in 1 mutation, 2 mutations, 3 mutations, and 4 mutations, all in different types of that gene. All mutations inactivate the gene. And so and now I'm in the process. I only have 10 billion cells that I've looked at, which is wussy in this field. I wouldn't publish this until I had probably 10 to 100 trillion. But so then I can take these mutants that I know exactly what they need to do to evolve, and I can ask them to evolve. I can put them in a medium where if they do evolve, I will know overnight. Because the selective advantage of being able to make, in this case, the amino acid tryptophan is so enormous that I will find that out overnight if that happens. And so I can ask, what happens when you need 2 mutations and you only get an advantage when you have both? At this point, the answer is nothing. And that is actually supported by the literature. What's different about this is I am specifically asking these questions. Most cases, these are things that people discover kind of on the side. You don't do experiments to test the limits of evolution. And in particular, my work is designed to actually test that.
How many would you say are working in the area that you're working in right now?
There are a few people. It's sort of boring research because until things evolve, we get really excited when things do happen. But they're not. I'm probably a little fish in a little pond at this point.
Well, my understanding, I think you mentioned there was a scientist that's been running an experiment now for 30 years.
Well, he's been 35,000 generations. To my knowledge, he began this in the late 80s, so we're talking 15 years. That's Richard Langsky at Michigan State.
And has he been able to demonstrate the development of new function where you have to have two steps?
One of the problems was he didn't ask a very interesting question. His microbes are evolving in a very low glucose. They're better at it. Most of what they got better at, that happened in 2,000 generations. So he's been watching very, very slow advances since that. And 2,000 generations is your first year. So he's been looking at very, very slow advances since then.
We have your PowerPoint.
Oh, good. Wrong one. I'm sorry.
Wrong one.
That's that's a lot longer than.
So.
It was testimony.
I see.
No, it's okay. There's one good picture. Other than that, it's you can just close it out.
So you just close this.
Close it out. Yeah, it's not that big a
Okay.
deal. So the point is.
Here we go.
PowerPoint testimony of Ralph Silke, P8. Yeah, there it is. That one.
This one right there.
Yeah, there it is. This is not really. It's not really all that it's cracked up to be. Whoa, same one. Just close it out. There's one. That'd be it. That's it. Yeah.
Okay.
Yeah, so then keep going. Yeah. So these are some examples of where evolution has worked. You have, for instance, if you have, you can put 10 million bacteria that are able to use the sugar alcohol ribitol. Some of them will be able to produce an ability to grow on xylitol. The last example is one that has been batted around in the discussions on intelligent design. There is an evolved beta-galactosidase that is actually is both as an example of evolution and an example of when you ask it to do two things, failure happens. Barry Hall made a name for himself with the evolved beta-galactosidase, and he put that next slide up. Lactose use. This is a system where if you cut out the original enzyme for lactose use, there is an enzyme hiding in E. coli that allows it to use lactose. However, you will never find this, ever, unless you cheat. And the way you cheat is by allowing lactose to come in. And if you cheat, if you artificially stimulate that cell to allow it to have lactose come in, you will get mutations. But if you ask it to do two things at once, you will never see that happen. And so it's an example of both evolution and inaction of evolution.
What is? Can you put in perspective the conclusion that evolution can take one step? If one step will produce new function, but your work is suggesting it cannot take two steps. What is the significance of that?
Well, the significance of that is simply all over the place. There are all sorts of things that you don't get any advantage to the organism until both things happen. Simple matter of using a sugar. To use a sugar, you got to do two things. You got to bring it in, and you got to use it. If you can bring in a sugar and you can't use it, you are not helped. If you can use the sugar but you can't bring it in, you're still starving. And Michael Behe has made the flagella a classic example of this. Actually, the flagella is worse than what Behe says, because if you're able to move and you don't know where you're going, you don't have an advantage either. And there's an entire other system that allows a bacteria to tell where it's going. That it's got sensors hooked up to the motor that allow it to figure out where it's going. And so it's worse than what Behe has made out to be with the bacterial flagella. But there are large, all over the place, there are systems where nothing happens unless you have multiple events taking place. And to my knowledge, that is the state of experimental evolution. So when you are saying you are producing new things by macroevolution, it's a leap, an enormous leap. You have not shown that. And as a matter of fact, the evidence is to the contrary.
At that point, what I'd like to do is, how does what you're talking about relate to the proposed changes in the minority report? And I'm putting up on the screen right now a reference to the proposed change in the indicator that would have students understand the difference between micro and macro evolution. And I guess I would ask you whether this suggested additional specificity under paragraph D is appropriate, and is it appropriate to make a distinction between micro and macro?
Sure. I would obviously agree that I think microevolution is interesting. I think it's people should understand it. You can do cool stuff evolving bacteria, but they should understand the large difference between being able to do that and being able to produce new body forms, or the other, even the other rather modest steps that would be needed in a macroevolutionary scenario.
Well, then, the work you're talking about, the biological experiments, involve bacteria.
Right.
And is that actually you're working in a microevolutionary paradigm?
Right.
So
exactly.
You're actually not testing macroevolution.
It's hard to test. But in theory, I am asking if macroevolution works by microevolutionary steps. If macroevolution is simply the extension, then knowing vigorously what microevolution can do will tell us what macroevolution can do. And right now, 2 steps seems to be a block.
And since Michael Behe's publication of his work, Darwin's Black Box, which discusses the theory of irreducible complexity, there's been a lot of scientific debate over his concept. Are you familiar with that?
Yes, I am.
And what would you say the current status is? Has his idea been refuted? Is it still being debated? What is the status?
Well, I think it's, unfortunately, not as many people have gone to the lab as I would have liked them to have gone to the lab for this sort of work. I think, and that was one of my frustrations. Behe and Ken Miller would get on public radio, and Behe would say, "It can evolve," and Ken Miller would say, "Can too," and Behe would say, "Can not," and Miller would say, "Can too," and I'd think, both you guys go to the lab and start doing some work. And so that sort of inspired me to this. So now Lenski has essentially failed to produce a lot in bacteria, and he went to digital organisms. And he has shown that in digital organisms, as long as each step is selective, you can get some very interesting things. But yeah, he's gone to digital organisms, which are basically computer models of microbes, and has shown that as long as each step is advantageous, you can do things. So, which I think we knew before.
Is it fair then to say that while microevolution has been observed in the laboratory, macroevolution has not and is simply inferred,
Yes, I would say that's an accurate statement.
and the inference is drawn from what?
Well, the inference is, I think, you start with the assumption that nature is all there is, and this is all we have to work with, and therefore we're here, and there's nothing else that could have gotten us here, so therefore it must have worked, even though it doesn't look like it could work.
But you said you're starting with an assumption.
Yeah, you start with that assumption, and if you start with the assumption, which is what methodological naturalism does, if you start with the assumption that nature is all there is, you will produce an explanation that comes out of nature. And if it's a bad explanation, go back to your assumption. You won't work beyond that assumption.
This particular indicator, the second sentence says these kinds of macroevolutionary explanations generally are not based on direct observation and often reflect historical narratives based on inferences from indirect or circumstantial evidence. Is that a scientifically valid statement?
I think that's often true. It's right. The observations are more narrative, narrative as in speculation.
And I think you've answered this question. Can macroevolutionary explanations such as the formation of new body plans be directly observed either in or out of the lab?
Those are harder experiments to do. Now, in terms of body plans, in the field of developmental biology, there is a real effort to produce things. There are some very interesting mutations that have occurred. I would not say entirely new body plans, but some very interesting changes have been made. One of my favorite is there are fruit flies with legs growing out of their heads, and they're really cool. But that's not a real, doesn't help to have legs growing out of your heads. There are also...
so you would say that, in a sense, is macroevolution, but it is an evolutionary change that will kill the organism?
Well, these actually live. But they're not very good. They're not very good flies. So that's not a new body plan. That's probably the closest has been some geneticists have been able to add a set of wings to fruit flies. Unfortunately, that second set doesn't work. And that's as close. That's changes to the basic body plan. I would not call that a new body plan. Would be going from a worm to an insect.
The second set of wings, would that add to the function of the fly or reduce its function?
No. Having a set of wings that doesn't work is not a particularly good thing to fly.
Going to, I have on the screen here indicator 1A. Is that a scientifically valid addition to the description of biological evolution? Biological evolution postulates an unpredictable and unguided natural process that has no discernible direction or goal.
Yes, I think that's a very accurate statement of evolution, as it's typically, and it has to be, because if you're talking natural sources, natural processes, you can't have a guide.
And then the second sentence, it also assumes that life arose from an unguided natural process.
Right.
Okay, I'd like to move to the indicator indicator 6, subparagraph C of the additional specificity, where we're talking about natural selection, genetic drift, genomes, and mechanisms of genetic change provide a context in which to ask research questions and help explain observed changes in populations. However, reverse engineering and indirected thinking are used to understand the function of biosystems and information. Is that an appropriate addition, and could you comment on it?
Yes, I think that's actually, as someone who loves experimental science and who thinks that teaching is, if you can get kids excited about things, I think this is actually a very useful, I hate this word, but I'll use it, pedagogical tool. In the sense that if you tell students they can understand things by simply asking, well, if you were a bacteria, what would you do? And it turns out the answers that they come up with are reasonable and rational and right. That the microbe actually, one person says they're not intelligent, but they do the intelligent thing. If I want to understand a system, as a matter of fact, when I teach my students how bacteria use lactose, I tell them exactly that. What would you do if you were a bacteria? Well, if you've got lots of other sugars around and lactose is hard to use, I'd use that first. Well, that's exactly what the bacteria does. If you have a bunch of genes that are needed to make lactose, when are you going to turn those on? Well, let's turn them on when the glucose runs out. Good idea. And that's exactly what the bacteria does. And so, rational teaching, from a rational standpoint, and again, you don't have to claim that they're rational or designed. You can quote Dawkins if you want to, that things simply appear designed. But as a teaching methodology, this provides a very good handle for understanding systems.
There is another indicator. Before you pass that, you understand what methodological naturalism is?
Yes.
Well, would you say that in this one indicator where you're asking the student to look at the bacteria as if you were the bacteria, how would you do that? That's sort of a teleological or a design or end-directed. Is it fair to say that in that kind of methodology, you're actually using methodological design rather than methodological naturalism?
Yeah, that's probably a good description. And you can certainly simply say that this works. It works as a way of understanding it. If you're looking at the structure of a set of genes, if genes A, B, and C are all involved in the same process, if you find out where gene A is, where do you think gene B would? Well, if you were the bacteria, where would you put gene B? Well, how about right next to gene A? And it turns out that you're generally right. And so that approach, that's what scientists do. Whether they acknowledge it or not, they look at this and they say, how would I do this if I were the microbe? And the creative ones figure it out, do the experiments to test that, and they sort through the possible answers, and that's generally how science advances.
I have on the screen a indicator that would have students understand that the sequence of nucleotide bases within genes is not dictated by any known chemical or physical law. Is that a scientifically accurate and valid statement?
Yes, that's an accurate statement. And it has to be accurate. As a matter of fact, for years, one of the reasons the discovery of DNA as the genetic material was a late discovery historically was because we thought DNA was boring. We thought it had a very repeated structure, that it was holding the proteins together because proteins were interesting and therefore they had to be the genetic material. And because we thought because we thought it was repeated and boring, therefore it could have no information. If there was a law that says A had to be next to T, had to be next to C, had to be next to G, you would have no information. So in order to be an informational molecule, you can't have the order dictated by law.
And that brings up another question, is that indicator 1 of standard 7 benchmark 3 here, this is a description of scientific knowledge, and the majority report would have scientific knowledge describes and explains the physical world in terms of matter, energy, and the forces, whereas the minority proposes that we simply say scientific knowledge describes and explains the natural world, which in your view is the better statement?
Well, I think there's the glaring omission, in the definition of as it is information. And the other is, I think it would... this
You
is
mean biological information?
biological information, exactly.
And could you be a little bit more specific about what you mean by that?
It's the code. It's the DNA. It's the sequences.
The messages?
The message. The message is what matter, energy, and forces do not explain.
Okay. And so, since you cannot reduce science, if you did reduce science to just the physical world, then you would omit investigation of biological information.
Yes, I think that's one of the things. Or your understanding would be quite constrained. But it has to be approached as information.
Finally, we have about 10 seconds. Sequence of nucleotide bases within, we already talked about that. I think that's about it. Dr. Sielke, do you have any general comment on the proposed changes that are contained in the minority report?
I think you'll have a group of students who will be much more aware. They'll have a better understanding of evolution. They will not accept it unthinking. Those who accept it will have accepted it in light of the criticisms. So they will be stronger because of that. Those who doubt it will know why they doubt it, rather than simply having been told to doubt it. And I think you'll... now, this is a subject where being sure may not be the best thing you want. You may want people who don't know the answer, because that may be the better place. Not if you don't know something, and you know you don't know something, you're better off than if you don't know it and you think you know it. So I think you'll have a better educated population.
Thank you so much for your testimony.
Thank you.
Yes, I do believe that the Earth is 4.5 billion years old.
I'm glad to hear that. Sir, we have no questions for you.
This was fascinating again, and you lost me quite a ways back, but I tried to stay with you. And, the idea that you could do this with bacteria is just fascinating to me, and I applaud your efforts, even though you say you're not very many of you're a minority working in this field, but we think that you're a true scientist, and we really appreciate all the things that you're trying to do to keep science, doing the right thing and letting children learn what true science is doing. Appreciate it.
Thank you.
Well, I welcome you also, Dr. Sielke. Your closing comment gave me some confidence that I might try to attempt to converse with you when you said that as long as something like if you know you don't know, then you're in a good place. So I don't know a whole lot about this subject. I find it fascinating, and I really appreciate you taking the time to come speak with us and help us to understand.
Thank you.
Now, so let me see if I have this right. Basically, you use microevolution trying to create macroevolution. Or is it backwards?
Well, no, I think that's accurate. I think if I were to, if I could consistently show that that you can consistently, with bacteria, if you need 2 or 3 or 4 events, and you can get them to happen, I wouldn't be rich, but I might be famous. Because that would throw ID out the window. And I think and if that experiment were to work, it would throw ID out the window. And just to point out the importance of this, Barry Hall in 1991 thought he showed that bacteria could do 2 mutations, and that if he had a system very similar to mine, where nothing happened in theory until 2 things happened. Okay, and he thought he thought he was able to observe that in bacteria. That was in 1991. He got a paper in the Proceedings of the National Academy of Sciences out of that. Now that's a fairly prestigious journal, and so the point was, you could. Now I don't think it made the front page news. But it made a prestigious journal. In 1988, someone named Cairns thought he found a mechanism where bacteria, if a mutation was advantageous, that it would happen more often. Okay, so instead of being random, it was something that would happen more often. He also made big news in the scientific circles. So the point is that this is not. People may not want to invest their careers in it, but they find it interesting. And I think one of the things that is helpful, and this is why being skeptical of evolution, and this is where an attraction to design comes in, is the evolutionists don't ask these questions. Because if you know that, I am what evolution can really do, you don't ask bacteria to evolve because you know the answer right away. And so it's only when you step out of that paradigm and you say, well, maybe it doesn't have all the answers that you even ask the question. And so that's why I think the skepticism of evolution is good for science. Evolution needs an opposing theory because it'll just sit on its haunches and spin just so stories that everybody's happy with. And so that's why if you have someone from the outside saying, it ain't necessarily so, and let's do some experiments where other people are happy just with just so stories, you are helping science.
When you have a fruit fly that grows a second pair of wings that don't work, why is that not a mutation or a change? Why would one argue that's evolution?
Yeah, it's a mutation. Certainly, it's a mutation and you could call it evolution. It's a change with time. That one, if I remember correctly, and that actually took 3 steps, and that was a highly engineered change. I think the developmental biologists wanted to see if they could do it.
Okay, I'll rest my curiosities. Thank you.
Dr. Seelke, earlier today we heard that evolution is a rather slippery word. Therefore, when you're describing evolution in bacteria, what is happening? What would you be more specific about that word?
Oh, I would. What I am doing is microevolution. But again, if you make the assumption that macroevolution occurred by what we can see in microevolution, what I'm trying to do is vigorously push what we know microevolution could do. And again, if we can push that, that is how a plausible theory of macroevolution would come about. And what I see is, unfortunately, most people are just satisfied with the stories, that they don't want to push microevolution to find out what it could do. They're satisfied with saying, we take this, add several million years by the same thing, and out comes a macroevolutionary change. So I would say I'm doing microevolution with an attempt to understand what it can do, which has implications towards macroevolution.
Are you saying that the evolution in the bacteria that you are doing and the other experiments that you have been describing are actually adding genetic material?
What I would do, now the ones that I am doing, there is a small addition, right? There's an addition of capabilities that occurs. Now, for instance, if I take my gene and I have one mistake in it, okay, by random processes, that one mistake can be fixed, and I think you would say yes, that cell is better, and it has gained a little bit of information. It is This is a small gain that produces a very large change. The gene that I'm looking at has 268 amino acids. 267 of them are right. Okay? There's one mistake. There's one base change causing one amino acid change, and the thing is broke. And so, yeah, I'm making one change. Has that cell gained information? It would appear, yes, that it has gained information. Not a lot
I
of
would say-
information.
Okay, where I'm going with that is that when bacteria become resistant to antibiotics, I have been under the impression by reading and involvement with it that it is not necessarily an added amount of genetic material, but it is actually a dysfunction that allows those bacteria to become resistant and therefore multiply but it is not an actual added genetic material.
It's not a net adding, but it would be, I don't know, how would I compare it? It's like you have a word and it's misspelled and you fix the word. Have you added information by fixing the word? You still had 7 or 8 letters. So I think this may be semantics.
Time is up. There's others, but I thank you for your time.
Thank you.
Audible hopes you have enjoyed this program.